Hello everyone. DNA or deoxyribonucleic acid is the hereditary material in almost all living organisms. It carries the genetic blueprint of life. The length of DNA is measured in nucleotides or base pairs. For example, E. coli bacterium has around 4.6 million base pairs, while humans have about 3.3 billion base pairs in their haploid DNA. Let's recall the basic unit of DNA, the nucleotides. Each nucleotide has three parts: a nitrogen base, a deoxyribose sugar, a phosphate group. There are two types of nitrogen base, purines and pyrimidines. Purines having adenine and guanine. Pyrimidines are cytosine and thymine. In RNA, thymine is replaced by uracil. Nucleotides are connected through a 3' to 5' phosphodiester bond to form a chain. This creates a polymer with a 5' end having a free phosphate group and a 3' end with a free OH group. The backbone is made of sugar and phosphate, while the nitrogen base projects out from the chain. The double helix model of DNA was proposed in 1953 by Watson and Crick using X-ray diffraction data from Wilkins and Franklin. क्या दोनों DNA strands एक जैसे होते हैं? They are complementary, not identical. Thanks to base pairing rule, adenine pair with thymine, 2 hydrogen bonds, guanine pair with cytosine with 3 hydrogen bonds. This maintains a uniform distance between strands. Key features of the DNA double helix: 2 strands with sugar phosphate backbone, anti-parallel orientation, 1 strand having 5' to 3', while the other having 3' to 5', anti-parallel orientation. Right-handed coiling, 10 base pairs per turn with 0.34 nanometer spacing, and the base and hydrogen bonds provide stability. क्यों हमेशा एक प्यूरिन एक पायरिमिडिन के सामने आता है? Because this pairing ensures uniform spacing across the helix, that essential for structural stability. This complementary nature of DNA allows accurate replication. Each strand can serve as a template to produce identical daughter DNA molecule. क्या यही genetic inheritance का कारण है? Yes, this structure supports genetic inheritance and expression. The Central Dogma of molecular biology describes how genetic information flows. DNA to RNA, RNA to protein. The process of formation of RNA from DNA is called transcription, and the process from which proteins are formed by RNA, this is called as translation. अगर RNA से DNA बनता तो at that time this process is reversed, that is formation of DNA from RNA. This is called as reverse transcription. The process of formation of DNA from RNA, this is called as reverse transcription. that's seen in some viruses like retroviruses. Retroviruses का एग्जाम्पल आप ले सकते हैं HIV. What we have studied, why DNA matters. DNA is more than a molecule. It is the script of life, carrying codes that define what we are and how we function. यही कारण है कि DNA की स्ट्रक्चर और वर्क को समझना एसेंशियल है, not just for biology student but for every interested in life science.